Mr. J, a 23-year-old male, has been suffering from a dry cough for 2 months. He's prescribed diphenhydramine 25 milligrams each night by his doctor. The patient returns in a week and notes that he's feeling a little bit better, but the cough is still there. The doctor then increases the diphenhydramine dose to 50 milligrams, but the patient retreats to lower the dose after 3 days of use because of intolerable morning drowsiness with the higher dose.
2 weeks later,
the patient returns complaining of the same symptoms after 2 weeks, so the physician switched him to cetirizine 10 milligrams. Mr. J fills the prescription and returns to his physician after 6 months for follow-up, telling him that he's feeling now much better on the new medication. While this typical clinical scenario involves some effort to learn from experience, the approach is rather not really organized and can be improved upon, hence introducing N-of-1 trial. Randomized controlled trials remain on top of the throne for clinical research. I know it said N-of-1 trials in the title. We'll get back to this in a bit. If a new therapy succeeds in an RCT, then it can be used in the future for people with the relevant condition. But RCTs have a problem, and this could affect any one of us in the future. Why? By nature, RCTs measure average effects. So, on average, a person will benefit from a medication that passes in RCT. The distribution in RCTs represents the difference in group means, so we can divide it up into a placebo and a treatment group. If a positive effect was demonstrated, it means that the treatment group had more people who responded better to the treatment on average. But the more doesn't imply all. It's still very possible to have some people who did not respond to therapy, or even some that have been harmed by the treatment. So even when a therapy is shown to work, there is still possibility that it will not work for a small part of the population, or that another drug might work better in certain people. It is true that RCTs are considered the gold standard for proving that new medications are effective for the majority of the population. But if you want to show that a therapy works for an individual, how do we get around the problem of the average effect? Well, epidemiology has a way, and that's the topic for our video. In this video, we're going to tell you more about the N-of-1 trial.
The N-of-1 trial gets its name from the fact that only one person participates in the experiment. They're also known as single-case designs for personalized studies. So the N-of-1 trials are double-blinded, randomized, crossover trials with a limited number of patients and most likely as small as a single patient. It can be used to determine if an existing therapy is conferring the intended benefit or also used for a trial of a new therapy. In an N-of-1 trial, the patient and the provider are both typically blinded to the order in which the therapy is administered. But how come a whole study is done on one person while RCTs need hundreds or thousands of people as participants? In an N-of-1 trial, they use crossover between an active treatment and a placebo, which means that the person starts with one treatment, spends some amount of time taking it, and then the person will switch over to the other treatment. The N-of-1 trial design is repeated for as long as needed, sometimes with washout periods in between treatments. In an RCT, many people provide only one observation, but in an N-of-1 trial, one person provides many observations over time. So the patient acts as their own control, and then the researcher conducting the trial just needs to analyze the difference between the days where the patient was taking treatment A and the days where he or she was taking treatment B. And what else is also amazing about the N of 1 trials is that they're considered to match the gold standard of evidence set by the RCTs. What does a gold standard of evidence means? RCTs allow us to make causal statements somehow about therapies. They allow us to say that a treatment causes a specific change to the disease. And just like RCTs, N of 1 trials allow us to go beyond this correlation and prove a causal mechanism between the treatment and its effects. However, there's a key difference here. Unlike RCTs, N of 1 trials study individual treatment effects, also known as evidence that a therapy works in a single individual. And because of this type of design is useful in the field of personalized medicine.
But if they're that useful, why aren't they used more? Individualized treatment effect is extremely useful if you happen to be that one person, but not so much for everyone else. Obviously, it has a generalizability problem. Pharmaceutical companies are the ones producing new drugs. They want to make sure it works well enough for a wide enough market to be profitable. Therefore, are N of 1 trial profitable for pharmaceutical companies? I don't think so. Since we need to follow someone over time, possibly a long amount of time, N of 1 trials are best suited to chronic diseases. It's also better if the treatment works immediately. A fact that makes N of 1 trial really cool is that they shine in the arena of rare diseases. For people with these orphan diseases, it's unlikely that an RCT will help them directly. One example of a published N of 1 trial using anti-seizure drugs. In this study, they assessed the efficacy and safety of dazinamide in 15 patients with focal seizures. Mix of patients with focal aware and focal impairment awareness seizures experiencing at least 4 seizures per month despite phenytoin use. After determining each patient's dazinamide dose, patients were randomized to 6 5-week periods in pairs of dazinamide or placebo. At the conclusion of the study, the investigators were able to demonstrate a significant decrease in seizure frequency of 37.9% using 2 statistical procedures. Think about this. N of 1 trials don't need to be held by large hospitals or pharmaceutical companies. You can start with today just by using your phone or even just a pen and paper. Nothing can stop you from gathering data on your own and using an N of 1 trial design to figure out how to improve your life. N of 1 trials can change individual lives.